A very good evening to everyone. Hello dear students, I am Bushra, a botany faculty, and today I am going to discuss the questions which are mistaken by most of the students in this paper, which is AIATS Plus Phase 2 Test number 8, which was held on 9th of March 2025. So in this session, I am going to take the questions from these specific topics. First two chapters, that is sexual reproduction in flowering plants and organisms and population, are from 12th class and they carry very much weightage from exam point of view. And the next four chapters are from class 11, that is plant kingdom, cell the unit of life, photosynthesis and higher plant, and morphology of flowering plants. All the four chapters are very important from exam point of view. I know this time is quite lethargic from complete day, but I will make sure that you take some great knowledge from this session. So let's sprout some new ideas and branch out your knowledge. Let's start with the very first question, which is mistaken by most of the students. Let's see the very first question, that is question number 102, and it is from the chapter Cell the Unit of Life, and it says centrioles differ from axoneme of cilia or flagella. So from this question I can see that it is somehow talking about the cytoskeleton or the cytoskeleton से क्या क्या चीजें बनी होती है उसके बारे में. Let's see that how it goes. So let's start our class. So what is cytoskeleton? Okay, let me change the color of my pen because it is not visible. Right. Okay, so the first one is cytoskeleton. Cytoskeleton basically composed of some filamentous substances which help in providing the framework of the cell. जितना भी framework या फिर structure होता है cell का, वो किससे होता है? Because of the presence of cytoskeleton. And it is present in the eukaryotic organisms. अगर हम इसके functions की बात करें, it helps in the mobility, in providing the structure, etc., maintaining the structure of the cell. Now let's see what are its components. Right, so it is basically have three major components, or we can say it is composed of three different types. Let's see. So the first one is microtubule. Next one is microfilament. Although these things are not given in the NCERT, I'm just telling you that how it works and how it goes on. And the third one is intermediate filament. So if we talk about first one, that is microtubule, it is basically composed of tubulin proteins. These are cylindrical structures, cylindrical and hollow. Cylindrical and hollow. अगर हम functions की बात करें, so it helps in the cell division or spindle formation. Functions, I'm writing FXN for functions, so it is basically help in the spindle formation that is very much required during the cell division, right? That's why it is very important to learn that what microtubules are. Spindle fiber formation that is required for cell division. Next one is microfilament. So these are composed of certain proteins which are known as actin. Along with that, one more protein which is known as myosin. These are cylindrical structure, but they are solid. They are not hollow as of like microtubules. Okay. If we see the functions, that what are its functions, so it provides the structure or help in the structure framework of plasma membrane. Plasma membrane को support करता है ये. And one more function, it helps in the pseudopodia formation. Pseudopodia, which is the structure formed in the amoeba. Right? तो वहाँ पे जो filament होता है वो होता है microfilaments. Next one is intermediate filament. It is composed of acidic proteins and these are cylindrical, hollow. So out of 3, 2 are hollow, 1 is solid. If we see the function of intermediate filament, it basically forms the scaffold of chromatin material, okay? And it basically forms the basket around the nucleus. So if we see the function, it forms basket-like structure or the scaffold, a complete जैसे ढांचा सा होता है वैसे ही न्यूक्लियस के चारों तरफ वो बना लेता है. So it's look like a basket around nucleus. So this is all about the cytoskeleton, right? Now let's move to the organelle, which is non-membrane bound, okay? So the non-membrane bound organelle, please comment down which are the organelles which are non-membrane bound. Okay, one is ribosome, another is centriosome. Let me choose the color, another one is centriosome. So centriosome are non-membrane bound organelles which are present in eukaryotic organism. अगर हम eukaryotic organism में बात करें, they are present in fungi, algae, in lower plants but absent in higher plants. Okay, they are present in animals, etc., but they are absent in higher plants. Please remember this point. Right? Non-membrane bound. Like this. Okay, and it is composed of 2 centrioles. इनमें 2 centrioles होता है and those 2 centrioles are arranged perpendicular to each other. I am putting sign for here. For that, as they are present perpendicular to each other, if I draw image for that, it somehow look like this. These are 2 centrioles, somehow look like perpendicular to each other, and they are surrounded by one amorphous substance, cloudy-like substance, which is known as centriosphere or gynoplasm. Okay. So this is the structure for the centriosome. If we talk about 2 centrioles, what do you call when 2 centrioles are present? Right? Now let's move ahead with the structure, that how actually the centriosome look like. So let's start from the very center of the structure. If I make the central structure, which is somehow made up of proteinaceous substance and it is known as hub. From that, 9 radial spokes are radiating out like this. Okay, there must be 9 radial spokes. Please count for me that they are 9. So yes, they are 9. And at the periphery, they have fibrils. And those fibrils have 3 subfibers like this. These are subfibers. These are made up of tubulin proteins, that means microtubules. Okay, like this. Arrangement उनका कुछ इस तरीके से होता है. Okay. And if we could name them, it is like A, B and C. A सबसे पास में जो है radial spoke के वो mentioned है, and the last one is C. Right, so this is how somehow they look like, and now how these subfibers are attached to each other. They are attached like this from CA linker protein. Don't worry, I am going to label them all. CA linker protein like this. Okay. I think there is some issue from the comment section, as I am not able to see any comments yet, but it's okay. Let's move ahead in the session itself. So they are connected with each other from a Ca linker, and they look like somehow like cartwheel. So अगर पूछा जाए कि cartwheel appearance किसका होता है, then it is centriole. Okay? अगर हम number of microtubules count करें, it have 9 peripheral microtubules, but in the center there is no presence of microtubules. That's why the arrangement is 9 plus 0, like this. If we see the surrounding, it is not at all surrounded by any sort of membrane, but it is surrounded by one amorphous substance which is known as pericentriolar satellite or mesiole. I am writing here that centriole is surrounded by pericentriolar satellite, और उसको हम mesiole भी कहते हैं, और mesiole. अगर हम इसके पेरीफेरी की बात करें तो ठीक है. Now please count for me that how many microtubules are present in the periphery of centriole. So as I've told you that fibril proteins are present in the multiple of 3, that not in multiple of 3, sorry, for in the 3 subfibers. They have 3 subfibers A, B and C. That means total number of microtubules which are found in the centriole is 27. How? 9 into 3, 27, right? Because there are 9 peripheral fibril proteins. This is all about the centriole. Now, what are the functions of centriole? So, if we see what are the functions of centriole, it forms the basal body for cilia and flagella. Basal body for cilia and flagella. Cilia and flagella क्या होता है? These are the locomotory structures which are found in the prokaryotic and eukaryotic organism. And what is the function of centriole? It helps in the spindle fiber formation during the cell division. Spindle fiber formation. So these are the function for centriole. Right, let's move to the structure of cilia and flagella. So somehow the structure of cilia and flagella are similar. Let's see how. Here you can see that the complete structure is given. Let's start from the center again. So these two are central microtubules. Central microtubules. Again, it is covered by one sheath. This is one sheath which is known as central sheath. The centrally located microtubules are covered by central sheath. Here you can see there are 9 peripheral microtubules which are in doublet. दो के number में है, right? And they are 9. So these are peripheral microtubules. Right, and those peripheral microtubules are connected to the central sheath via radial spokes. So these line, ये जो आपको line दिख रहे हैं, these are the radial spokes. Right, now let's see that these are doublet, and these doublet are again, here you can see that if I name them like A and B, so these are attached to each other via dynein protein, right? And the proteins which are present, these white-like structure, this balloon-like structure which are present here, these are nexin proteins which are attached to the peripheral microtubule doublet. So if we see the arrangement of microtubules in the cilia and flagella, it come out to be 9 plus 2. How? There are 9 peripheral microtubules which are in doublet. That means The peripheral microtubules are in number 18, and the centrally located one, the singlet one, are in number 2. So if someone asks you that the total number of microtubules which are present in the cilia and flagella is 20, please don't get confused between that how many peripheral microtubules are present and how many single microtubules are present in the cilia and flagella structure. Right? So if we see the previous slide, in this we have seen that this is the structure of centriole and it is not surrounded by any sort of membrane. Apart from that, it is surrounded by one amorphous substance, or we can say the cloud-like substance, which is known as pericentriolar satellite or mesiole. But if we see that in the cilia and flagella, it is surrounded by plasma membrane. And this structure which is enclosed by the plasma membrane, it is exonyme. So अभी तक जिसकी बात करी, this 9+2 arrangement, etc., how many microtubules are present at the periphery, how many microtubules are present at the center, all these we are talking about the exonyme of cilia flagella, which is the core of cilia and flagella. Okay. Likewise, there is one statement or content. Oops, I think it is not clear. Let me erase the content for you so that you can see clearly. Yeah. Okay. So here one content is written in the NCERT which states the difference between the cilia and flagella. If we see the first one, the cilia and flagella are hair-like outgrowth of the cell membrane. Yes, obviously it is completely correct. It is the similarity between them as they are the locomotory structure. Next one is cilia, small structure which work like oars, causing the movement of either the cell or the surrounding fluid. So it is one of the major difference between the cilia and flagella that the oar-like movement is done by cilia, not by flagella. And the next line which says the flagella is comparatively longer as compared to the cilia and responsible for the cell movement. Yes, if we see the size, cilia हमेशा बहुत ज़्यादा in number में होते हैं and they are smaller in size. But if we talk about the flagella, they are quite longer as compared to the cilia, but they are few in number. Next, the prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. So these are the some differences or similarities between the cilia and flagella. Now, let's get back to the question which says centriole differ from the exonyme of cilia or flagella. Let's see the differences or the statements given in the question. First one is a number of microtubules present at the periphery. तो अगर हम periphery की बात करें, so the number of microtubules present in the centriole is different. The number of microtubules which are present at the periphery of centriole is 27, but we talk about the exonyme of cilia and flagella, it is 18. So yes, it is one of the difference between them. Let's move ahead with the next one which says in presence of radial spokes. दोनों structures में 9 radial spokes present है, so it is not the difference between them. Next one is in arrangement of microtubule. तो अगर हम centriole की बात करें तो वहाँ पे arrangement of microtubule था 9+0. But if we talk about exonyme of cilia and flagella, it is 9+2. So yes, it is one of the difference between the centriole and the exonyme of cilia and flagella. Next one is in containing proteinaceous central hub. So central hub किस में था? In centriole, but not in exonyme of cilia or flagella. So ये भी एक difference हुआ. Next one is in being surrounded by double membrane. So if we see the structure, centriole में तो कोई membranous like structure नहीं था, but exonyme of cilia and flagella में एक plasma membrane थी, but वो भी double membrane bound structure नहीं था. तो ये दोनों के लिए false है. Okay, so out of the 5 features, 3 are true for them. Those are A, C and D, and it is correctly depicted in option 1. So this is how we hit the correct answer for the question. Let's move to the next question, that is question number 94, which says Freudian starch is the stored food in. As I can see that this question is from the chapter Plant Kingdom. It is quite large chapter. Let's see what we can study. That Freudian starch is the stored food. So let's talk about the stored food material in different algae. So if we see, there are different types of algae, So algae are classified into 3 different classes on the basis of their pigment. Let's classify them. So the first one is, let's make it one classification like table. So the first one is chlorophyceae. Chlorophyceae नाम से ही पता चल रहा है, that means it will be somehow look like chlorophyll like color, which is green. Next one is phaeophyceae. इसके नाम से तो पता नहीं चल रहा, but its common name is brown algae. Third one is rhodophyceae. Rhodo, red start from the R, and the same this name also starts from the alphabet R, so we can see that the common name is red algae. So if we see the cell wall composition, so the inner cell wall, the inner side of the cell wall in the chlorophyceae is composed of cellulose, inner side, and if we see the outer side, it will be composed of pectose, right? And if we talk about that how they reproduce, so if we see the vegetative reproduction in them, I am writing VR for that, then asexual reproduction AR for that, and sexual reproduction SR for that. This is common for all the 3 classes of algae. Let's start with the very first one, that is vegetative reproduction. That means how they are going to reproduce vegetatively. So their mode is fragmentation. And if we talk about the asexual reproduction, they reproduce by biflagellated zoospores. And if we talk about the type of sexual reproduction they follow all the 3 types. Those are isogamous, anisogamous, and oogamous type. Right? In chlorophyceae, one more feature is present. They have the presence of pyrenoids, and they store food in some sort of oil droplets also. What pyrenoids are? Those are proteins beside the starch. It is generally asked in the NEET exam. So please remember that what pyrenoids are. These are proteins besides the starch. I am writing here for you. Pyrenoids, protein beside starch. Let's see the next one, which is phaeophyceae, and the common name is brown algae. अब इनको brown algae क्यों कहा जाता है? Because of the presence of fucoxanthin or xanthophylls, due to which they appear or exhibit the range of olive green to brown color, right? That's why they are known as brown algae or phaeophyceae. As we know that algae generally small होते हैं, but if we talk about the phaeophyceae, they are profusely branched. They are profusely branched. For example, ectocarpus. और ये कुछ इस हद तक बड़े हो सकते हैं, for like 100 feet. For example, kelps. Right? If we talk about their cell wall, so the cell wall have deposition of algin. I think this color is not visible to you. Let me change. Light green. If we see the body structure, that how they actually look like, so they have some structure which help in holding the substrate, that is hold fast. So if this is the algae, this is the hold fast for that. And there is one stalk which is known as stipe. And there is one leaf-like structure, photosynthetic structure in the Phaeophyceae, which is known as frond. So this is how their complete body is differentiated into three structures. So if someone asked you that stipe, holdfast and frond are found in which class of fungi, so you can say that it is Phaeophyceae, right? And if we see, okay, let me take one page, it is completely, okay, discard, oops, इसको हम, let me put them on the, like this, okay. Where is my content? I think it is somehow mistakenly I have deleted. I'm very sorry for that, but I think this video is recorded, so you can go back and see whatever I have written there. So where I am actually about at the Phaeophyceae, that how Phaeophyceae works and what are the things which are present in the members of Phaeophyceae. And there we are talking about how they are going to reproduce. So if we talk about the vegetative reproduction in them, in Phaeophyceae, let me write again for you. Phaeophyceae, it is fragmentation. This fragmentation is going to be common in all the three classes. If we see the asexual reproduction, it is from zoospores, biflagellated zoospores. One very interesting biflagellated zoospores are present because the gametes which they are produced will be of pyriform shaped. Sorry, it will come under sexual reproduction, not in asexual reproduction. Right. So if we talk about the sexual reproduction, they will produce the gamete which are pear-like shape and they are biflagellated. So this is about the brown algae or Phaeophyceae. Next one is Rhodophyceae. Rhodophyceae, the common name is red algae. That means they appear red in color, right? So if we talk about their how they look like, they have very complex body organization as compared to other two classes. Complex body organization, right? And if we talk about their vegetative reproduction, again, they have fragmentation. Through that, they reproduce vegetatively. If we talk about asexual reproduction, they reproduce via non-motile spores. The motile stage is absent in members of Phaeophyceae, please remember that. And we talk about the sexual reproduction, they are reproduced via non-motile gametes. That means they have oogamy or oogamous type of sexual reproduction, right? So this is all about the Rhodophyceae, Phaeophyceae and the Chlorophyceae in the previous slide. Now here one very beautiful image from the NCERT itself, that how we can remember all the major features from these three classes of algae. So here you can see the common name is given for the classes of algae and their major pigments. How you are going to remember them? So in the major pigment you can see that chlorophyll A is common, and after that start writing B, C and D. This is how you are going to remember all the major pigments in the different classes of algae. Next, if we talk about the stored food. So here you can see the starch is stored food in the Chlorophyceae, and if we talk about the Phaeophyceae, the brown algae, in which it is the complex carbohydrate, which is in the form of mannitol or laminarin. right? Now let's see the next one, which is rhodophyceae, in which Floridian starch is the stored food material and it is structurally similar to amylopectin and glycogen. Please remember this, amylopectin and glycogen. So they are structurally similar. This Floridian starch. Okay, and cell wall, again you can see that cellulose is very much common in all the three algae. Difference came here, algin, which I have told you that cell wall का deposition in the members of Phaeophyceae, cell wall में algin present होता है. Next one is pectin and polysulfate esters are present in the cell wall of Rhodophyceae. Next is, if we talk about the number of flagella, so the most number of flagella are found in Chlorophyceae. They could be either 2 to 8 or more than that. And in the next one, the Phaeophyceae, 2 unequal laterally attached flagella is present. As I have told you, they have biflagellated zoospore in which the flagella are unequal in size. If we talk about the gametes, which are pear-like or pyriform shape, those are also having 2 flagella, right? And the feature which is very important from exam point of view as well, that in members of Rhodophyceae, the number of flagella are absent. That means that they lack flagella in their complete life cycle. Okay. If we talk about the habitat, the members of Chlorophyceae are found in freshwater, brackish water, salt water, and the Phaeophyceae are found in freshwater, brackish water, salt water, etc. And same with the Rhodophyceae. So this is how we are going to remember the complete features for the algae, different classes of algae. Okay, let's get back to the question. So question number 94, which says the Floridian starch is the stored food in. So it is the stored food in the members of Rhodophyceae. तो अब हमने examples तो याद किए ही नहीं, right? The Chlorophyceae के example होते हैं Eleutheryx, Chlamydomonas, Volvox, etc. So these are the examples which are generally asked in the NEET exam and Spirogyra. If we talk about the brown algae or the members of Phaeophyceae, those are Ectocarpus, Sargassum, Fucus, etc. Right? So these are the example for green and brown algae. And if we talk about the red algae, those are Gelidium, Gracilaria, Porphyra, right? So these are the example for the red algae. Now let's see the options. The first one is Ectocarpus. It belongs to the brown algae or Phaeophyceae, in which the stored food material is laminarin or mannitol. If we see the Eudorhina, it is also the brown algae. Third one is Fucus, it is also the brown algae. Last one is Porphyra, which is an example for the red algae, in which Floridian starch is the stored food material. Therefore, here we hit the correct answer for the given question. Let's move to the next question, that is question number 97, which is from the chapter Morphology in Flowering Plants. Let's see the question, which says Syncarpus Gynoecium is or are found in. So if we talk about the Gynoecium, which is the female reproductive part of the flower, right, and carpel Gynoecium is the female reproductive part of the flower and carpel is its unit. Right? अगर एक से ज़्यादा कार्पल हैं, they can be in the form of fused state or in free state. Right? अगर हम बात करें fused की, then it is known as syncarpus. If we talk about when they are in free, then it is known as apocarpus. Right? And if we see the question, it is asking about the Syncarpus Gynoecium, that means fused one. Let's see the examples. The first one is mustard. So mustard belongs to the family Brassicaceae and they have Syncarpus Gynoecium. That means those are fused. Next one is tomato, which belongs to the Solanaceae family, and again it has the Syncarpus Gynoecium. So again, this is the correct example for the Syncarpus. C is lotus. They have apocarpus. That means carpels are free. And the last one is rose. Again, in them, apocarpus, gynoecium is present. And here we can see that out of 4, 2 are true. And those are given in option 2, which says only A and B. This is quite easy question. Let's move to the next question. That is question number 100. Which is from the chapter Cell, the Unit of Life. As I've told you, it is one of the most important chapter from exam point of view, and from this chapter a lot many questions are asked, about 4 to 5 questions, right? So it is very important. Let's see the statements. The first one is, in plants, cell wall protects the cell from mechanical damage and infection. And statement B, which says the middle lamella glues the different neighboring cells together. So the concept is basically based on the cell wall of the organism. So cell wall is the rigid non-living structure present outside of the plasma membrane, and it is present in fungus, plant and algae, but it is absent in animal cell. हम में cell wall नहीं होती, right? Now, if we see that the fungal cell wall, it is composed of chitin. The cell wall which is present in the fungus is known as chitin, and its subunits are N-acetyl glucosamine units. Okay, and if we talk about the plant cell wall, it is composed of cellulose, the major part, then galactans, mannans, and some minerals. Next is algal cell wall, and it is composed of insoluble polysaccharide, which includes hemicellulose, pectin and protein. So these are the different cell wall composition. Please try to remember all these terms with respect to their particular plant cell, particular organism type, because this is very important from exam point of view. Now, so question is asking about the cell wall features, right? So if we talk about the plant cell wall, the major concern is, as I am teaching you botany, so major concern is plant. So let's see that a plant cell wall, they could be of 2 types, okay? So the first one is primary cell wall. जैसे कि नाम से ही समझ में आ रहा है primary, that means it could be present from the very beginning of the life of the plant. जो सबसे पहले आता है वो primary है, okay? Next one is secondary. Okay, so the next type is secondary type, secondary cell wall. Secondary cell wall, as नाम से ही पता चल रहा है, it comes later in the life when the maturation in the plant arises, right? Now let's see the plant cell like this I am making. It is composed of primary cell wall, and again another cell of plant is like this. So this is how 2 plant cell will look like when they are put aside, right? So it is composed of first primary cell wall, and then where the secondary cell wall deposition will start. It will start towards the inner side. It will start towards the, it will start towards the inner side like this. And at maturity, the primary cell wall will start diminishing and secondary cell wall will take place its role. राइट जैसे जैसे प्लांट वुडी होता जाता है वैसे वैसे उसकी प्राइमरी सेल वॉल डिमिनिश होती जाती है और सेकेंडरी सेल वॉल से सारा डिपॉजिट होता है. दैट्स व्हाई इट शोज द वुडी लाइक स्ट्रक्चर ड्यू टू द डिपॉजिशन ऑफ सेकेंडरी सेल वॉल, राइट. एंड अगर हम लोग सेल वॉल के बीच की बात करें, सो दिस इज प्राइमरी सेल वॉल, द सेल primary cell wall of 2 plant cells, and the region which is present between the 2 cell wall of the 2 plant cells, it will be occupied by middle lamella like this. This region, this green region is occupied by middle lamella. And middle lamella is composed of calcium pectate and magnesium pectate, right? As I am repeating again and again that the comment section is not able to move properly. I don't know, there must be some problem. The previous comments from the previous session are still appearing. But let's see, let's wait so that I can reply your queries, what you are writing in the comment section, and might be you are answering them also. Okay, so this is middle lamella, which is composed of calcium pectate and magnesium pectate. Like, this is actually the gluing agent between the two plant cells, or we can say the cementing layer between them. Okay, let's get back to the question and the statement A, which is saying that in plants, cell wall protects the cell from mechanical damage and infection. So yes, it is absolutely true that the main function of the cell wall is to protect the cell from any sort of damage. So yes, it is absolutely correct. Next one is the middle lamella glues the different neighboring cells together. Just now we have seen that how that this middle lamella is gluing the two different plant cells together. So yes, this statement is also true. So here both statement A and B are correct. Let's see the next question, that is question number 101, and it says again from the chapter Cell, the Unit of Life, which says membrane protein can be classified as integral and peripheral based on the. अब integral और peripheral protein हमने कहाँ पढ़े थे? It is from plasma membrane. So this is the structure, the beautiful structure, the fluid mosaic model of plasma membrane, which is given by Singer and Nicholson. Okay, let's see that how it actually is, that why it is known as phospholipid bilayer. Okay. So it is basically composed of two lipids layer. How? This is how it look like. One hydrophilic lipid and the tail is towards the inner side, and from the other side this hydrophilic and tails are hydrophobic like this. So this is the complete structure, how it goes, as you can see here as well. These round structures, these are hydrophilic structures, hydrophilic polar heads, and these tails are hydrophobic. So these heads are basically, they are protecting the hydrophobic tails from the aqueous environment. Okay, ये plasma membrane है, तो aqueous environment से घिरा हुआ है, and these heads are actually protecting the tails. And here we can see that these are the integral proteins which are present. The proteins which are present inside the membrane, वो पूरा अच्छे से घुसा रहता है. These are known as integral proteins. And how they are classified? They are classified on the basis of their ease of extraction. Protein जितना अंदर ज़्यादा embedded होगा in the plasma membrane, उतना ही उसको दिक्कत होगा निकालने में. That's how we segregate them or classify them. Next one is the peripheral proteins. So these are the peripheral proteins which are present in the plasma membrane. Right, these are quite easy to extract, जैसे कि ये periphery पे attached है. Now here you can see that some proteins are attached, and on proteins these carbohydrate molecules are attached. These yellow structure, these are carbohydrate moiety. If this carbohydrate moiety is attached to the lipids, if it is attached to the lipids, for example this green one, if it is attached to the lipids, then it is known as glycolipids. If it is attached to the proteins like this, then it is known as glycoprotein. इन सब के अलावा प्लाज्मा मेम्ब्रेन में कोलेस्ट्रॉल भी प्रेजेंट होता है. If we see the plasma membrane of erythrocyte, particularly human RBC का plasma membrane, then if we see the composition of lipids and proteins in them, then the proteins come out to be 52% and lipids come out to be 40%. Please remember these terms because, please remember these value because it is very much important from exam point of view. Again, 52% protein and 40% lipids in human RBC membrane or erythrocyte. Okay, so this is the quasi-fluid nature of the lipids. So if we talk about the quasi-fluid nature of lipids, which enables the lateral movement of proteins within the lipid bilayer. Okay, that is quasi-fluid nature of lipids. So if we see what could be the functions for the plasma membrane, so the functions, let me change the color. So the functions for plasma membrane, it could be the endocytosis, secretion, cell division, etc. These are the functions that what plasma membrane could show. And the major function is transport. Plasma membrane hai, it must be providing certain transport through inside to outside of the cell. Obviously एक लाइन बनी हुई है, एक गेट बना हुआ है कि सेल के अंदर क्या सामान आ सकता है और क्या सामान बाहर जा सकता है. So major function क्या हुआ? Transport, right? And they are semi permeable in nature. Semi permeable होती है plasma membrane, right? And transport could be of two types. First one is passive. नाम से ही समझ में आ रहा है. That means they do not require any sort of energy to transport the material from one side to the another side. And the next one type is active. So if we talk about the passive type of transport, in one type where we require no sort of proteins to carry the material from inside to the outside, and the next one in which some carrier is required. So in passive, there are again two types. In one type, the material will transport from one area to the another area according to their gradient, according to their concentration gradient. Okay? Concentration gradient के accordingly particles move करते हैं. Next one is they required certain types of carrier proteins so that they can transfer from one side to the another side. For example, there are certain polar molecules which cannot pass this plasma membrane. So they must require certain carrier molecules so that they can pass the membrane. Okay, so this is facilitated. And the type of transport where the concentration gradient is required, the name is diffusion. Yes. Sorry, I forgot to mention earlier. And when we talk about the water, the diffusion of water across the membrane, one special term is given for that, that is osmosis, especially for the transport of water across the membrane. Right? Now, if we see the active transport where energy is required, उसका बहुत ही beautiful example है sodium potassium pump. अक्सर ये एग्जाम में भी पूछा जा चुका है, so please remember that sodium potassium pump require energy. To transport the sodium or potassium ion across the membrane, we require energy in the form of ATP. Right? So this is all about the plasma membrane, the structure, etc. So the question is about the membrane protein, which is classified as the integral or the peripheral based on the, so as I've told you, there are two type, integral or peripheral. जो embedded है और जो periphery में present है, obviously on the basis of ease of their extraction, they are classified. Rest all options get ruled out. First one is saying difference in their chemical composition. Obviously, composition is going to be same in them. Next one is surface charge distribution. There is no surface charge as such present in them, and difference in size. Size doesn't matter here. Okay. So from this, so the answer of this question is option 2. Let's see the next question, that is question number 107. Right, and this question is from the chapter Photosynthesis and Higher Plants. So here we have to mark the following statements as true or false and select the correct option. So here certain statements are given, as I can see that those statements are particularly from the Calvin cycle, or we can say the C3 cycle. So let's start with the concept of Calvin cycle in the next slide. C3 cycle or Calvin cycle. C3 cycle क्यों कहते हैं? Let's see. Calvin cycle तो इसीलिए कहते हैं क्योंकि it was discovered by Melvin Calvin by using C14 Okay, by using C14, the heavy isotope of carbon in green algae. और वो green algae कौन सी थी? It is Cladophora. If I am not wrong, it's Cladophora only. So this C3 cycle is actually universal, or we can say it occurs in all the photosynthetic plants. Occur in all the photosynthetic plants, irrespective कि वो कैसा भी है. It is basically composed of three major steps. First one is carboxylation. Next step is reduction. And the third step is regeneration. So अगर हम लोग C3 साइकिल की बात करें तो इनमें हमारे ये तीन स्टेप मेजर हैं. And out of these three steps, one is one of the most crucial step for the process of Calvin cycle or C3 cycle, that is carboxylation. Please remember that. Again, I am telling you that it is very important from exam point of view. So why carboxylation is very much important? क्यों ये बाकी दोनों steps उतने important नहीं हैं, and why it is considered as the most crucial step in the C3 cycle. So Calvin cycle है, it is one of the cycle or phase in the dark reaction, right? Carboxylation, जैसे कि नाम से ही पता चल रहा है, that means fixation of carbon dioxide, which is very important. हमको कार्बन डाइऑक्साइड को फिक्स करना है so that we can make the carbohydrate substance. Okay, so our sir is here. Sir, please look that I am not able to see any of the comments yet because the comment section is, I think, freezed for the previous slot in the screen which is just in front of me. Okay, let's proceed with the session. So here fixation of carbon dioxide takes place. तो इस fixation को करने के लिए one enzyme is required, that is Rubisco. The most abundant enzyme on this planet is Rubisco, which is ribulose, bisphosphate, carboxylase and oxygenase activity. Here as it is fixing the carbon dioxide, so here it is showing the carboxylase. Here it is showing the carboxylase activity. Okay. Next is reduction. Let's see all these things in the diagram itself. So here that carbon dioxide along with water is fixing via the process known as carboxylation by the action of enzyme Rubisco, and this is done it is done by RUBP. This is the 5-carbon compound, right? And it is get combined with the carbon dioxide, again one more carbon, so that यहाँ पर 6 carbon का compound बनना चाहिए. But instead of that, here we can obtain 2 molecules of 3-carbon compound, that is 3-phosphoglycerate. So this 3-phosphoglycerate is actually 3-carbon containing compound. And the first stable compound of this cycle is phosphoglycerate, due to which it is known as C3 cycle. अब समझ में आया that why it is known as C3 cycle? Because of the first stable product of this cycle is 3-carbon containing. Now next one is reduction step. अब जो यहाँ पे ये बना phosphoglycerate, it will get reduced. For reduction, it will require some amount of energy, and the energy will be provided in the form of ATP. Along with that, NADPH will also be required, right? So ATP is utilized and get converted into ADP and NADP, right? For reduction step, for each molecule of carbon dioxide, and in reduction step, 2 molecules of each ATP and NADPH are required. Please remember this value as well because it is very important. अभी आगे पता चलेगा कि क्यों. ठीक है, तो यहाँ पे 2 molecules of ATP and 2 molecules of NADPH are required in the reduction step. Next, what is doing? It is making one triose phosphate which will further form certain molecules in the sucrose or the carbohydrates molecules, and the rest will again get regenerate. For regeneration, again, energy is required in the form of ATP. Remember, regeneration में NADH नहीं require हो रहा है, सिर्फ ATP हो रहा है और वो भी एक molecule. Okay? So regeneration will again regenerate the primary acceptor of the carbon dioxide of C3 cycle, that is RUBP. So regeneration is very much important for the cycle to continue uninterruptedly. Okay, so here each and every step is very much important. अब अगर हम देखें and summarize this problem, and अगर हम देखें and summarize this complete cycle, the first one is that here we are going carboxylation, that means fixing of carbon dioxide and then forming the 2 molecules of 3 carbon compound, where we have used the 2 molecules of ATP, and here we have used 1 molecule of ATP during the process of regeneration. That means total 3 molecules of ATP and 2 molecules of NADPH which is required during the reduction step. स्टार्टिंग में हमारा जो CO2 था वो कितना था? It is only 1. That means for fixing a 1 carbon dioxide molecule, we have to use 3 molecules of ATP and 12 molecules of NADPH. अगर हम ये एक ग्लूकोज की बात करें, for one single glucose molecule जिसके अंदर 6 कार्बन होते हैं, that means to fix the 6 carbon molecule, that means glucose जिसके अंदर 6 carbon है, that means इस cycle को 6 times होना पड़ेगा. For that we will multiply this value with 3. That means to fix 1 glucose molecule, 18 ATP and 12 NADPH are required. Please remember this value as well. अगर हम single molecule की बात करें, 3 ATP 2 NADPH, we will multiply it by 3, 18 ATP and 12 NADPH for 1 glucose molecule to fix. Okay? Let's see the question. Okay. So the first one is regeneration of RUBP is crucial for Calvin cycle to continue uninterrupted. So yes, it is very much required क्योंकि RUBP जो कि primary acceptor है Calvin cycle का, and one more thing, RUBP is 5 carbon compound. उसके वजह से इसको C5 नहीं कहेंगे, उसका first stable compound जो है वो 3 carbon का है, that's why it is known as C3 cycle. Okay, so yes, it is very much important step. So this is correct statement. So from this I can rule out option 1 and option 3 as they are claiming that statement A is false. Next one is plants adapted to dry tropical regions are mostly C3 plants. So अगर C3 plants की बात करें तो इनका जो temperature optimum काफ़ी कम होता है. It is not present in the dry tropical regions. These are generally found in the temperate region जहाँ पे थोड़ा temperature normal रहता है. In the tropical regions, C4 plants are present. Due to time constraint, I am not able to explain you C4 cycle, but hopefully in the next class. So this statement is incorrect. Okay. Next one is Calvin cycle is common to the C3 and C4 plants. So yes, it is absolutely correct, as I have told you that C3 cycle is universal, or we can say that it is found in all the photosynthetic plants. So here statement A and C are true, which is correctly depicted in option 4. Therefore, for the question, rest all options get ruled out and the correct answer is option 4. I hope this session will help you to strengthen your concepts. So students, please keep yourself healthy, okay? And spread the colors like pollens and happiness like oxygen. Happy Holi, stay hydrated and stay fit. Thank you for watching this session.